Hi, my name's Shelley and I'm a microbiologist at the Synthetic Biology Research Centre based at the University of Nottingham, and today we're going to talk about microbes. So microbes are tiny living things, so small that we can't see them with our eyes, and they're everywhere. They're in water, in soil, in the air, and even in our bodies. There are different types of microbes. Bacteria, which is what we work on at the university, viruses and fungi. Microbes can be good, they can be bad, and they can be extremely useful. So today we're going to take a look at a useful microbe called yeast. So yeast has been around for years, and it's been used for bread making as far back as the Egyptians, over 5,000 years ago. They thought that whatever made bread rise, or the little bubbles that you see, was magic. In 1857, a scientist called Louis Pasteur discovered that it was yeast was responsible for fermentation. So fermentation is when yeast converts sugars into carbon dioxide and alcohol, and that causes little bubbles, and that's what makes your bread rise. So we're going to do an experiment with yeast that you can do at home. So what you're going to need is 4 bottles, 4 balloons. It's important to make sure there's no holes in the balloons. You're going to need some yeast, some sugar solutions, and some water. So you need your water to be warm but not too hot. So this is just nice, you can put your hands in it and it's not gonna burn you. So the first thing we're going to do is we're going to put a teaspoon of yeast into each of the bottles without getting it all over the floor. Okay. And then we're going to add our sugar sources. So this first bottle here is going to be our control, so we're not going to put any sugar in there. This next bottle, I have some glucose that I got from the lab. You can use any sugar you can find in your kitchen. Granulated sugar, caster sugar, whatever you can find. And we're going to put a teaspoon of that into this second bottle. Okay. Now, into this third bottle, we're going to put a little jelly teddy bear, and into this last bottle, we're going to put a sweetener. Just going to put the lids on these so that I don't knock them all over the floor. And the next thing we're going to do is we're going to put some water into each of these bottles. So you don't want to fill it right to the top. There we go. So the next thing you want to do is give these a bit of a shake. So it's a clever person would have kept the lids. I didn't, so I'm going to use my hand. Just want to give them a quick shake just to make sure that everything's nice and mixed. And the next thing we're going to do is we're going to put our balloons on the top of the bottles. So, like that. So there we have our control, our glucose balloon, our lovely jelly teddy balloon, and our sweetener. So now we're just going to leave those for 20 minutes and let's see what happens. Okay, so it's been left for 20 minutes. Let's see what happened. So with this one, our control, we can see there's absolutely nothing in the balloon. So this is showing us that without sugar, the yeast can't ferment. As you can see in this one, which is our glucose one, we have a nice full balloon. So what's happened in here is the yeast has been able to use that sugar and produce carbon dioxide, which you can see is all these little bubbles up here, which has sort of escaped into here as well. Now this one, our jelly sweet, hasn't blown up very much, but you can see, if we give the balloon a little bit of a squeeze, that there's definitely some CO2 in there. Now why do we think that might not have worked as well as this one? Well, if you remember, the sugar that went into this went in as a powder, so it was easily dissolved in the water. The sugar that went into this is the jelly sweet, so that sugar is all bound together, which means that the yeast can't actually utilise it as quickly because we've got to wait for that sweet to dissolve in the water before it can actually be used. Now this one, this is our sweetener, and you can see there's absolutely no reaction that's happened here. There's no CO2 being produced. So you can also tell there's no bubbles in the mixture. Do we have any idea why that might be? So if we think about what a sweetener is, so sweeteners we use as a replacement to sugar, so they don't actually have the sugar in there, which means that the yeast can't use those as an energy source. So that's what's happened there. So at the Synthetic Biology Research Centre and the Carbon Recycling Network, which we're part of, we use other microbes such as bacteria, which also undergo fermentation, and they produce loads of useful chemicals that can be turned into really useful things like biofuels. So if you want to know more about that, you can come and have a look at our website, which is sbrc-nottingham at ac.uk or carbonrecycling.net. Thanks for watching, and I hope you enjoyed doing the experiment.